Good afternoon to everybody who decided to spend 1 hour and a half of this afternoon with us at Sano at our seminar. Well, I think that we have already something like 20 registered participants. It doesn't mean that there are only 20 persons attending the seminar, because many are present here, sitting in rooms where more than one person, and so I think that we can start our seminar. And after break, which was due to, first of all, due to Easter, and then due to a conference on high performance computing organized by Frontnet, which was this time really quite large, with 221 participants. But well, we have more than 20 participants here, so it's great. And today we have two speakers. The first speaker is Artur Blat. Artur is at that moment a PhD student, and as a PhD student, he is employed by Kraków Technology Institute. This Kraków Technology Institute is a member of research network Łukasiewicz. And of course, Artur is affiliated as a PhD student at the Jagiellonian University. And he has earned his Master of Science degree in biochemistry in 2022. So probably Artur is almost at the end of his PhD research. And the second author is our colleague from Sano, Adam Sułek, who is a postdoctoral researcher in the team Structural and Functional Genomic. 2022 was a very important year for both of them, because in this year, Adam obtained his PhD degree in chemistry from Jagiellonian University. So, what they will present here is a joint research in the framework of Sonata project of National Center for Science in Poland. So, as usual, if you have a question which requires immediate answer to understand the talk, please ask it on question and answers or on chat. I will try to transfer this question to our speakers. And of course, there will be time for question, answers, and discussion after the talk. So, Artur and Adam, screen is yours.
Thanks for introduction. And actually, this presentation will be divided into two parts. I will cover the first part with computational part, which is related with drug discovery, and it will present our take on different strategies. And actually, AI is no longer any novelty in this domain, it's a standard. And second part will be more into lab experiment, which is something new for most of you, so maybe it will be more excited. And this work is under consortium, under NCN grant. The grant is Sonata and related with combinatorial antimicrobial therapy in diabetic foot infections. Yeah, I will start with some limitations of AI models. And with this quite provocative title, there is no such thing as a validated prediction model. It's not my title. I find this and really resonate with domain of molecular design because yeah, you've probably all of you been there where your model has great metrics and scores of predictions, high ROC, low MSE or MSE, maybe some other metrics in drug design. This kind of metrics is a proxy of energy, how molecule binds to molecular target, for example, docking score. And some new PhD student discovered that some compounds could have better scores than known drug, and they think it is something very useful, but reality is quite different. So, and this limitation comes from a training data set, because if we go out from and we want to test examples which are far much more dissimilar to our training data set, you see that the error on the right side diagram, it increased the error. So, of course, one thing is that keep in mind that metrics are not everything. You have to check whether your model could be useful for this task. But another point of view is that what we can do to increase the variability of training data set, to find the new points, to build more robust model. And today I will give some hints how we deal with it. And there is something additional. There is even worse than that, because sometimes model has great scores, but very limited explanation. For this reason, we published, the work is under review, but hopefully it will be published soon. And we built a benchmark data set of 200,000 labeled molecule for XAI, for explainable AI. So what we usually want is check what input tools by model could be interpreted as the most important for classification or for prediction. And rule-based approach of pharmacophore from structural biology is approach in which we have 3D arrangement of some chemical features, and as I mentioned, it's purely rule-based approach. So according to these biochemical rules, we build the data set in this way that we match the hundred of or even million of compounds. And if a molecule not attached to these spherical points, of course, it's negative. If matched, there is a positive candidate. But sometimes molecule could match in more than one settings. And we remove these candidates because of unambiguity of this kind of label. So it allows us and others to compare whether their model predict according some rule-based method according pharmacophore explanation. And we checked the most famous models. One is molecular fingerprint, which divide molecules into smaller parts. And surprisingly, this kind of model has the best predictions in many domain. This is very simple model. It can be XGBoost or other tree-based method with these fragments, and usually it's a standard that these methods outperform more complicated methods. But in case of explanation, this model is worst. And we compare this with attention model, attention between each atom with each atom. So this kind of model could see the long range interaction. And finally, the conclusion from this work is that if we expect that two different aromatic rings are responsible for activity, then how model will give them the importance score. And for this fingerprint, the model don't recognize correctly which aromatic ring are responsible for the fitting into pharmacophore, but with this task, this attention-based model are quite better. But still, the results show that in molecular drug design, explanation of the model is still very limited. And another obstacle, even the most important, because if we have some results from the lab and we see that established benchmark has some activity scores or metrics, and after years it appears that some active compounds are present in many different assays. What is assay? I mentioned about how we design drug, and assay could be also designed because we need some molecular target, we need environment for this target, for example, different solvent, temperature, pH, and also we need some method which can read the readouts. So for example, fluorescence and absorbance spectroscopy can be misled by fluorescence signal of molecules, also called pains, which gives a signal but not specifically. This compounds not binds according mechanism we expect, but gives some false positive signal, and others disruption phenomena can produce some artifacts. And from our studies could be something like bad solvent and aggregation of compounds could be present and then binds and inhibit the enzyme, but not specifically. And this is exactly what we observe in our case, because in antimicrobial therapy, beta-lactamase enzyme is very important. This enzyme could lead to antibiotic resistance because it makes antibiotic therapy less effective. Simply, the used antibiotics are eaten by this enzyme. So in our approach, we want to inhibit this enzyme and makes this combinatorial therapy with antibiotics more successful. And in PubChem, the largest database of existed experiments, they are two different assays. And after years, it appears that one assay has much more active compounds, one order of magnitude more active compounds. So most researchers prefer to use this because they have more variability of active compounds, and of course for your model seems to be good. But the small details was that detergent was not added into this assay, and it leads to this aggregation what I mentioned. So that most of these compounds was false positive. So building this model in our case has very limited number of active compounds, especially compared to number of inactive compounds. And there is even worse because in these active compounds we have to remove compounds which creates covalent bond with which is related with antibiotic resistance also. So we want to remove this effect from our model. And finally, we compare this data set from PubChem with commercial available compounds from enamine company. And it showed that this chemical space is almost covered. And then we realized that this molecular target, beta-lactamase, was very important. So this company have some collaboration to find the best drug candidates here. But anyway, we decided to build some models to find something new. And this is good from point of view of applicability domain, which I mentioned, because these compounds are from the same chemical space. So we built different models, some neural networks, some tree-based methods, and we asked some data from other literature, set some threshold of activity, which is not usually the threshold in this case, when we have small amount of active compounds, we want to increase this number and find anything which is somehow active. So finally, the 100 candidates, and then we used molecular dynamics and molecular docking to select the best 7 candidates. And in next part, Artur will mention about this. So we use this ligand-based and structure-based approach to select 7 candidates. But next part, we select another 11 candidates, but with another approach. To screen this, because enamine has also offer 1 billion compounds, so 3 order of magnitude bigger purchase compounds which can be ordered to screen them in the lab. And to screen the billion candidates, it's no longer possible without some estimation and only checking of the most promising candidates. In our case, it could be a few months for screening, and using active learning in this case reduce the times of this task more than 10,000. And each prediction of this built model has some uncertainty. So the prediction is not only one point, but we have a whole distribution of different prediction. So what usually people are doing with in active learning is taking the most promising candidates, which is also called upper confidence bound, UCB, because we take the most promising candidates with the biggest uncertainty, and in each iteration we select the best, probably the best binders, and in each iteration this uncertainty is smaller and smaller. So after a few iteration, in our case after 10 iteration, we have more robust model, and probably with the best drug candidates, which can we estimate on this docking score, that after few iterations we have the best candidates, and it is quite common nowadays approach. And the details of this work will be presented soon in MCSB conference. But we can treat this problem even in 2 or 3 or more dimension. So this uncertainty of the model could be correlated. So the bigger uncertainty along one direction could be correlated with uncertainty with another target, and then this UCB approach works correctly because we optimistically take this mean prediction with some error along one axis and error across second axis, and the acquisition of this point will be taken from most promising candidates. But what if this prediction of 2 different targets are uncorrelated or negatively correlated? Then this selection will be inaccurate, and our preliminary studies show that our approach is better in terms of selection of the most promising candidates. So previously in 1D, of course, it was only one metric. Here, the metric which we calculate is hypervolume under this curve, and here there is Pareto front of most optimal. This red dots is the most optimal solution overall, the most global solution, and we check how fast our approach and other approach compared with random checking outperforms this in multi-optimization problem. So yeah, virtual screening is a great tool. Of course, predictive model, as I mentioned, is a standard. Could be very useful, especially active learning. We could say it's quite cheap and scalable, but as long as you have some computational power. So some part of this work is under AI factories project. So we have some access to supercomputers. We apply also for another computational grant, Euro HPC. And why it's not everything, because usually this why we want to have some high throughput screening experiment, and usually it is because imagine that some available data, of course, are published, but before they are published, the best drug candidates are patented. So if you want to have first access to best candidates, of course, you have to do your own wet lab work, because, as I mentioned, shared data are good for building some models, but it does not contain any unpatented candidates. So here I will present few methods which we are planning to use in the future in terms of this high throughput screening in the lab. One method is DNA encoded library. So this method are built from different building blocks. Here are presented as some blocks, but in reality they are some chemical compartment. So after each step, we are adding some identifier. This identifier is DNA code attached to this molecule, and in each cycle, next step, we are adding some different blocks with some identifiers. So finally, after a few iteration, each molecule, final molecule, has some identifier as a DNA barcode. But this method has some limitation. First one you can see is here, that this long and big linker of DNA could bind also to enzyme or protein, which means that it could gives you the false positive signal. So again, the same problem. But the idea here is that some company could build really big library for screening, even hundreds of millions. And finally, if some small molecule binds into protein, then it could be decoded with this DNA barcode. What was this molecule? So in one big or huge compound pool with million of molecules, after adding some protein, could be identified which the most promising candidates. But another limitation is high cost of this. Company could produce this, but it's more than most scientific grants budgets. So usually academia project has to think something has to think about something else. And we think that fluorescence polarization is then good alternative. Lower cost of instrumentation, lower cost of synthesis, especially, and still it has quite good sensitive and selectivity. And it works in this way that this tracer has some fluorescence probe, and it binds to the pocket according mechanism we want to observe. So if we add some pool of compounds which binds better than this, the free tracer in the solvent reduce the signal of fluorescence. And this method is much more sensitive than previously showed normal fluorescence without polarization. So it seems to be very useful even in terms of complex mixtures. So we have similar effect to DEL, but without this high expensive instrumentation. And this combinatorial synthesis is according some chemical rules. We cannot combine in any order, but according to some rules. And it is the same like in previous method, but you can see there is no barcode of DNA. So the question could be raised how we will identify which molecules in the pool works. But this is a part of the proposed grants. So maybe I hopefully in the future could tell something more how we plan to deal with it. So two of this project, first Team Feng and NCN Sonata, are a combination of active learning and experiments. And we are planning to perform up to 2100 of these experiments with lab automation. So we can compare that. In current project, we select 18 candidates and check them. So in this kind of project, you want to perform really high throughput screening. And third project is proof of concept. It's purely computational, but the common point with other projects are active learning. So it seems like active learning could be useful, especially in terms of reduce the amount of experimental work and high expensive computation. And two of these projects are in interest of commercial partners, Rivy and Molecule One, which support these grants, support the proposal. And to sum up this part, you have to remember that some metrics of prediction model are good, but still you have to check whether your model knows something about this data. So applicability domain is one, and another one things to check. Of course, these strategies contain not only AI and machine learning, but traditional structure-based analysis, docking, molecular dynamics, and so on. A combination of these methods give you higher chance to find something interesting. As I mentioned, active learning seems to be useful. It will not produce better scoring, but it reduces the number of experiment. And lab automation also seems to be not any longer optional, but it's more standard for this kind of project. And in the next part of the presentation, we will see something that one of compounds find with our approaches is under patent preparation and hopefully will be very useful in the future. So now I will give voice for my colleague.
Adam, maybe we can have questions to your part, not discussion, but just if there are some questions, maybe it would be good to answer them. What do you think about this?
Yeah, sure.
So if you have a question to what Adam presented here, please ask this question now because this part will be slightly different. So if not, we will have time to discuss it together after the end of the second part. Well, I think that there was some information about time used for processing in this learning AI process. Well, it was for your example, or it is from some literature?
Yeah, actually, it was from our approach. It depends how people defined it. In our case, it was more complicated. What is the most robust is if you have access to every solution. For example, for 2 million compounds, you calculate metrics and then you try find out the best candidates in these candidates. So you know the final results and you checked how many calculations do you need to reach there. So in this 2D and 3D, we did this for 2 million. So we have all of the final label. And usually it took less than 1% of whole calculation. But for this bigger data set, 1 billion data set, you don't have the final result of whole space. So it's kind of estimation how much calculation you will take, and you believe that according to some metrics, it seems that it will be hard to find something better. So more or less, it gives the final most effective results. So yeah, and in the literature, the range is similar, up to 1 or 2%.
Yes, and even it is on top of this slide about the data that they are sometimes this data are not reliable. So, do you think that one may some let's say improvement in this area, or simply it will stay as it is? And what are the main reasons that why these data are not so reliable?
Yeah, so if you have many different data sets, for example, for cancer research, for I don't know, heart attack or cardiovascular drugs and microbiology drugs, and it appears that the same compound is active everywhere. So the first sign that probably something is bad. And especially if you have the same instrumentation for finer readouts. So then you can see something could be wrong, and probably people who are responsible for this in the lab, if they observe that this kind of compound gives positive signal anyway, it should be removed. Yeah, so from if people do some analysis of the data and they see that the same type of compound it's quite often active, it should be removed. But in our case, if we plan in the next grant proposal, it will be better to just remove them before
It doesn't move, yes.
implementation. So
Yes, because otherwise this conclusion about what really was discovered is
exactly, it's like flipping the coin. You don't know, so you prefer to remove that noise signal.
Many thanks. I don't see any other questions on the list. So, as I understand, we will move. We'll move to Artur part this time. Yes. So, Artur, screen is yours now.
Okay. So good afternoon, everyone. My name is Artur Blat, and as it was mentioned, I am a PhD student at the Jagiellonian University. However, I am conducting my doctoral project in Kraków Institute of Technology, and my presentation is titled From Algorithms to Antibiotics, AI-based drug design for diabetic food infections. So as Adam was referring a lot to the bacteria antimicrobial resistance. So let's start with some facts about it. So according to data from World Health Organization, antibiotic resistance infections caused more than 1 million deaths globally in 2019. And they are this problem affects mostly low-income and middle-income countries. What is important that 1 in 6 bacterial infections are resistant to the antibiotic treatment, and it is a problem not only for human health but also for the economics, as it's estimated that that antimicrobial resistance can add around $1 trillion US dollars healthcare cost by 2050. Let's look at some data. These are the real data from the paper that describes what is happening currently in the Ukraine. Some soldiers with the battlefield wound infections were investigated, and bacteria were isolated from the wounds. And And as we can see, most of them, this six first of them belongs to so-called escape group of pathogens. And these pathogens are very important because they are very resistant and they are very often found in the wound infections. And it is also shown here on the right that these bacteria are multi-drug resistant strains. So they are not only found in the wounds, but they are also very resistant for antibiotic treatment, which is important for us and for the health. But how bacteria can deal with the antibiotics and what are the mechanisms of the antimicrobial resistance? So first of all, when you have the antibiotic, it can go inside of the bacterial cell and then it can be pumped out of the cell, but so-called drug efflux pumps. The antibiotic can be also modified. So it is modified by the enzymes that are inside of the bacterial cells, and it is not active anymore. Also, the antibiotic uptake can be inhibited, so it won't be possible for this modified antibiotic to go inside of the cell. But what is important here is that that some enzymes can can inactivate the antibiotics, and this is happening for beta-lactamase. I will describe in a moment. So we know from where the antimicrobial resistance come from, but how can we combat it? So there are different strategies. Most of them are, when we think about it, is antibiotics, but there are also a lot of different non-antibiotic antimicrobial therapies, like repurposed drugs. So these are drugs that are used in the clinics, but we found a different way to use them to treat different disease or infections, for example. It can be plant products as essential oils or plant extracts that have very often antimicrobial properties. They can be monoclonal antibodies, but also antimicrobial peptides that are secreted by the bacteria and also inside of the human body. It can be phage therapy and also nanoparticles. But what is important for us are adjuvants. And adjuvants are the molecules that more often don't have their own antimicrobial properties, but they can increase the effect, accelerate the effect of the other drugs, here antibiotics. So in combined therapy, the antibiotic works better. And this is important, as I will show it in a moment. And within our Sonata project, we work with the diabetic food infections. So in diabetes, persistent hyperglycemia can lead to nerve damage, which can increase food problems. And when we have these food problems, very often an ulcer can appear. And these ulcers are invaded by microorganisms where they can multiply and they can distract the tissue or accelerate the host's inflammatory response. These infections are often polymicrobial, meaning that we can find different strains of bacteria inside, and they involve very often antibiotic-resistant strains of organisms. organisms. That is why they can be difficult to treat. And the two main research objectives of these presentations are biological evaluation of AI-predicted beta-lactamase inhibitors and determination of the mechanism of action of one of them. And as Adam mentioned, one of the compound that we are working on is currently undergoing the patent application. So it was shown by Adam. We selected top 7 candidates which undergone biological evaluation. So what we did, we had a protein, beta-lactamase, and we tested the activity of this protein, treating the protein with different concentrations of our compounds, as they are designed to be beta-lactamase inhibitors. So they should act specifically on that protein and inhibit their activity. So as we see on the right, our compound, it had a IC50 value, which gives us the information how well it works. It was more than 300 micromolar, so it's not a potent inhibitor as we compare it with the well-known inhibitor of beta-lactamase avibactam, where the IC50 was 300 nanomolar. So we see that there was a difference of 1,000 times. But what happened was that then we investigated if these compounds that we are working on have antimicrobial properties. And interestingly, we found out that one of them has these properties. So we can divide bacteria into groups, gram-positive and gram-negative. And as we see here on this panel, our compound was working only on the gram-positive bacteria, because here, when we were treating the bacteria, gram-negative bacteria with different concentrations of the compounds, but there was still growth of bacteria. But here, when we look at gram-positive bacteria, we see in blue boxes that the growth was inhibited. And I would like to mention that the values here, that the first blue box that goes after the red ones is so-called minimal inhibitory concentration MIC, as I will refer it a lot. So, for example, we can tell that MIC value from Staphylococcus aureus was 32 micrograms per milliliter. So we knew that our compound has antimicrobial properties, and it kills gram-positive bacteria. But what exactly does it do to those bacteria? First, we performed growth. We performed a growth curves experiment when we treated our bacteria with different concentrations of the compound. And as we can see, when we compare our result to the control, which is blue line here, we see that the growth of the bacteria was lower compared to the control. But then, what exactly what exactly is the effect? Does it kill bacteria? It turned out that not really, because as when we compared our results from the time kill assay of the control to the samples treated with the compound, we see that when we added our compounds, it only inhibited the growth of the bacteria. So when we look what we had at the beginning and at the end, we see that the number of bacteria was not increasing, meaning that the compound does not have a bactericidal effect, it rather has a bacteriostatic effect. It is also shown, it can be seen here when we compare it to the positive control with bactericidal effect, which was ampicillin. So we see that the number of the bacteria decreased over the time, and around 8 hours, there was no live bacteria. And then we were wondering if this compound, if this compound will work with other well-described and well-known antibiotics. So we performed combination studies when we used our compound with different antibiotics, and surprisingly, we found that with one of them, meropenem, it works better in the combination. So when we compared the MIG value of the antibiotic meropenem and our compound alone, we see that the values decrease in combination. So the compound alone works with the MIG value 32 microgram per milliliter, but when we mix it with the meropenem, it was acting with the concentration of 8 microgram per milliliter. And from these values, we can calculate something which is called fractional inhibitory concentration index, FICI. And when we got the value which is half or lower, we can say that we have a synergistic effect. So what does it mean? That means that the combination has better effect than the compounds alone. And it is very important in terms of the antimicrobial resistance, because now when we, instead of using, let's say, 0.25 micrograms per milliliter of meropenem, we can decrease the concentration. So we are like starting to avoid the growing the antimicrobial resistance to this antibiotic. And that was confirmed for 2 strains of Staphylococcus aureus, for the strain that is not that resistant to the antibiotic treatment, which is called MSSA, and also for the resistant strain MRSA, on which the combinations worked even better. But that is also shown here in the time kill assay when we compare the effect of these 2 compounds alone and in combination. So we see that treating bacteria with meropenem alone, which is blue and red, was not acting effectively, because after time we still observed living bacteria. The same thing happened when we were treating bacteria with our compound. But when we use the combination of these 2 molecules, we see that after around 8 hours, the bacteria were killed and they remained dead until the end of the experiment. So yes, our combination, we found a combination of our compound with antibiotic that works with the synergistic effect, and that was confirmed here. But then we know that our compound works on the gram-positive bacteria, not on gram-negative. And what is the main difference between them is how their cell wall is built. So the cell wall is something that is around the bacterial cell, and there are differences in how they are built between gram-positive and gram-negative bacteria. That's why we were thinking that if it's only acting on gram-positive, it maybe affects the cell wall. So we treated our Staphylococcus aureus cells with the compound, and then we got the transmission electron microscopy images. And when we compare them, we see that the number of bacterial cells, which are shown here in like in circles, the number is like the same. They're almost the size of them is the same. They are still dividing, as we can see here in the middle of some cells. We see some line which indicates that they are still multiplying. So we didn't observe any effect on the cell wall. That was also confirmed when we treated our bacteria with the compound and performed a BCA assay, which allowed us to estimate the amount of the proteins outside of the bacteria. So when we compared the results of the treated samples to the control, we don't see any effect. So that means that there was no any protein leakage outside of the cell. So our compound is not affecting the bacterial cell membrane or cell wall, compared to the positive control, which was the bacteria treated with triton. Moving on, we also found antibiofilm properties of our compound. So we know that bacteria exist in two forms. They can be planktonic bacteria, and all results that I presented so far were for planktonic bacteria. And now let's move to biofilm. So biofilm is something that is formed, let's say, on a surface when bacteria can attach. And they multiply, they form like layers, they specialize, like different layers specialize in different activities. And it's important that the concentration of the antibiotic needed to disrupt this biofilm can be even 1,000 times bigger compared to the planktonic bacteria. So that is important also to study the biofilm and antibiofilm properties of the compounds. And as we can see here in the crystal violet staining, we see in the control there are small dots at the bottom of the well, and with the increasing concentration of the compound, we don't observe these dots anymore, meaning that the biofilm was completely disrupted. It is shown also on here on panel B, when we have the quantified data. So we see that in the half the concentration of the compound, the biofilm was completely inhibited. We can also see it there on the images from the confocal microscope. When we have the control, so this is biofilm, the green color indicates live bacteria. And then when we treat this biofilm with our compound, the red color starts to appear, indicating dead bacteria. But when we treat the biofilm with the combination of our compound with meropenem, the whole biofilm is composed of dead cells, and also it is disrupted, as we see there are some holes start to appear here. And it is important also to test the compounds on the cell lines, as we want our compounds to be working on the infections of the wound, so on the human body. So we need to know if the compound is cytotoxic against human cells. And as it was presented here for meropenem, we don't observe any cytotoxicity. And when we look at our compound, which is this dark green here, we see that it starts to be cytotoxic at the MIG value, which is 10 times MIG value. But what we are working here, and also in these computations, we are mostly working in the lower concentration. So, for example, we had the working in the combination with 1/4 of the mix. So these are still concentrations still safe for the human cells. And yes, as Adam mentioned, we first we had the 7 compounds, and these 7 compounds, the activity was tested against beta-lactamase, and the IC50 value was more than 300 micromolar. But then our AI team performed another, improved the model, and they selected another compound. And within this 11 new compounds, we see our progress. The IC50 for this new set of inhibitors is around 50 micromolar. So we see that our model is developing. It is working better and better, and the compounds that we are getting, that I'm testing in the lab, works better and better on the protein. So, short summary, we can see that the compound that I focused on was weak beta-lactamase inhibitor, but it was selective against gram-positive bacteria, and it had a bacteriostatic effect on them. We found out a synergistic effect with meropenem against gram-positive bacteria. We focus only on Staphylococcus aureus, these 2 strains. The cell wall or cell membrane were not disrupted. It had strong antibiotic properties, and it was not toxic at MIG concentrations for the human cells. And what are the future plans? So we want to further develop our models to get even better inhibitors of beta-lactamase. We want to determine the mechanism of action of this compound, specifically this one compound that we are working on. Because we still are not sure what is the mechanism of action, and we want to be sure. And then we want to also go further and test these compounds against clinical bacterial strains and on advanced biological models like human 3D skin models, and also perform some in vivo studies. And at the end, I would like to thank my colleagues, my research group from Łukasiewicz Research Center, my supervisor, Barbara Pucelik, and also our colleagues from Sano. And the research I described here was performed within the Sonata 19 project. So thank you very much, and if you have any questions, I will answer them now.
Artur, many thanks for your talk and presentation of this part of research. Well, it's great to have with us Olaf, because Olaf is always taking part in discussion after seminars. So Olaf, it's great to see you once again after your visit in Sano. Go on, please.
Hello. Yeah, I still have. I always have to wait until I can unmute here. So yeah.
No, it's great. Please go on.
So a couple of questions, actually. The first one, Artur, can you go to slide 3? I think there is an error in there. I'm not because I was looking for the Staphylococcus aureus, and you see that one at the last bar doesn't show any MDR strains, and there is E. coli, which shows 100% in that right diagram. So is that correct?
Okay. Yes, maybe I didn't explain it well. So these were the strains that were isolated from the wounds from the soldiers in the Ukraine, and within this, on the left we have the bacteria species that were isolated from these combat wounds, and then within this bacterial species, they were tested against their resistance to different antibiotics, and let's say like. We had like for Staphylococcus aureus, there were some strains isolated from these wounds, but none of them displayed multi-drug resistance. That is why there is nothing here, so 0, but only within this specific strains from these specific patients.
Okay, but E. coli being multiple resistance with 100%, that sounds a little bit odd, isn't it?
E. coli is also one of the pathogens that is good for human health, but when something goes wrong with the human body, it can be pathogenic. So
of course it can, but it shouldn't be 100% multi-drug resistant. Yeah, so that would be really disastrous if we would only have E. coli with multi-drug resistance, wouldn't it?
But if I remember correctly, from that paper, they isolated only like 2 strains of the. It was like very low number, so like
okay.
Within these two strains were multi-drug resistance. That is why it's 100.
Okay, thanks a lot to clarify that. I have one question also to Adam. So you're marking every compound, or no, you're not marking them. You're deleting those compounds that give ambiguous results. I've seen that in many other training scenarios for predictors. And what it typically leads to is that you have a predictor that always has full confidence, whether it's correct or not, because the ambiguous parts are completely wiped from the training set. Of course, there are parts of the, let's say, data space where it is ambiguous, probably because, let's say, the activity might be in a borderline kind of number of activity, but that would help you to also identify the spaces in the data space to say, okay, in this region, whatever we get as kind of a prediction is probably taken with a grain of salt. So you would know which part of your chemical space would be, let's say, unexplored area, perhaps because certain samples are missing. You could even suggest that some samples from this space should be tested, so in order to fill up the data space a little bit more. So I'm wondering, is that really necessary, or could you leave them in if you would, let's say, instead of classification, go for regression and just try to estimate a value instead of just active or inactive?
Yeah, so as Artur showed, in second iteration, we have better results for next iteration from completely new data set space for enamine, where our QSAR model doesn't work because the space was totally different than what we seen in PubChem. And in our case, it works better. And there we used structural biology like docking and molecular dynamics. Hopefully in the future we could use FEP also, but it's quite demanding in computational. But yeah, I would say that maybe not secret, but the crucial point was that using more structural biology gives us better results in this case. So I don't have much hope in standard machine learning model in domain where we don't have much data. Many classifiers in this domain has this additional information that be careful because it's outside domain of knowledge for this model. So yeah, the great scenario would be to have a lot of money in the project to test many alternatives. Yeah, but the reality in this kind of project is that 2 or 3 iteration with 10 compounds, it's everything what we have. So the improvement of the QSAR model, machine learning model, is not it's not big. Ideal scenario would be high throughput screening, as I mentioned, with 100 or 1,000 compounds tested, and it would be great in the future to have this financing and check this approach. That from one side we explore the new chemical space, and from the other we test what our model is predicting, because it's quite uncertain to just believe the final score of the model.
Okay, you said that you use docking than structure-based approaches in order to get at the winning compounds. So what did you use for docking? So there was nothing about the method here.
To be honest, we used two different engines. One is free available SMINA for active learning, and this big active learning pipeline we are using free software SMINA. But especially in this project, we used Schrödinger, so probably Glide.
Okay, I saw it, yes.
Yeah, and we also use GBS energy, which is more accurate than standard docking. So, and as I mentioned, it would be great to have this FEP in the future.
Okay. Yeah, there are a lot of drug binding force fields now that have been AI learned, and they seem to be at least on par with MMGBSA and sometimes even better.
Okay.
Thanks.
Well, I would like to encourage all present at the seminar to ask questions, to comment. Well, we have with us collaborators of both of Adam and Artur, from Sano and from Institute of Technology. So maybe you have something to add, which was not covered in this talk. Well, maybe when you are preparing for comment or ask question, I will. I think that it will be a kind a follow up of Olaf asked. Well, because I think that it would be good to have a kind of, let's say, a summary in which listed all tools which were used by you, and maybe besides of, let's say, conclusions on what you have achieved, maybe you have also some comments on usability, usefulness of these tools, because we are in such a stage where also not only practical results are important, but also I think a kind of evaluation of tools is also useful. So do you have some opinions about this?
Do you mean like from the computational part of?
From the computational part, yes, mostly from the computational part. But if you think that it's something it should be extended by other parts, why not? But for me, the most I'm interested in your opinion about these computational part tools.
Adam, would you like to answer the question?
Yeah, so I think I'm not surprised if something doesn't work, to be honest. Models, of course, when we are training the model, we want to have best score, best molecular input representation, and so on. But finally, it's biology, so many things has to be optimized. So in this case, how to prepare this essay, it's one thing that we've discussed. Sometimes things like solubility of compounds also has to be discussed before the purchase, because each compound costs like 200 euro for small fraction. So it's increase the cost. So we discuss, what do you think, guys, about these compounds, and some simple prediction model for toxicity. For example, Artur showed some data for skin cells. So we have to maybe not be so careful as cancer drugs in case of bacteria. But anyway, things like solubility, toxicity, and so on, we have to check and discuss. So it's so far it's this kind of interaction that any proposition and is discussed between teams. Yeah, and using different strategies, like structural biology really is not something obsolete. It has to be used, especially in domain of antimicrobial therapy, where the data are very limited, especially because pharma company is not interested in this kind of research.
Well, but I think that for all people, well, I'm not working in this area. I have never worked in this area, but I think that something like a methodology here, some remarks about this, about tools, I think that they, especially at that stage, they could be very, very important for many people. So, well, if I don't know to which extent my suggestion is reasonable, but maybe you could also, in the case of when you go for publication, maybe to present some results, some of your opinions about methodology, about tools. I think that
I think there is a lot of even blog posts. Some people prefer this, yeah, really, because it's a fast way, and everyone in the community could share their opinion. For example, I present quite high threshold when we treat molecules as active. So there are different approach. If people want to have better scores, probably they will change this threshold. For us, the most important thing is whether it will be working. So there are many different recommendations, and it depends from point of view. Yeah. So we have our approach, and probably during the publication process, some reviewers could have different opinion about the methodology. Yeah, so it's still under some different opinions, but it's good to track what other peoples are doing.
of course. Well, we have Barbara with question or comments. Barbara, please go on.
So just from the biological part and the general overview, we are really happy to see that everything is working so well, and we are very encouraged by the results. We're hopeful that our lead compounds that Artur presents will successfully pass patent protections and IP, as the data look very, very promising, and we hope that we can enter this compound to the clinic soon, after IP protection, of course. And we also hope for successful continued collaboration with Sano, because thank you guys, thank you for the collaboration, for the successful grant realization, and I'm really happy that we can work as a team.
Thank you very much, especially for your opinion about collaboration with Sano. Well, Sano is built also for collaboration with other units, because, well, nowadays science is a joint effort. So thank you. Well, I think that because we do not have more questions and we have almost reached the time devoted to this seminar, so I would like to thank Adam and Artur for very interesting talk, and many thanks. And I would like to invite all of you for seminar which will be next week at the same time. It will be seminar given by Luca Gerardini and Rosemary Blanco, and it will be something well between science and business, because the title of their talk will be From Neuroscience to Startup. So I hope that you will be interested in also in this process, but there will be a scientific and business component. So many thanks, and I'm looking for seeing you in a week from now.
Thank you.
Thanks a lot.
you.
Bye.
Thank you so much.